So we'll start with the production. So how to produce a monoclonal antibody? The name says monoclonal. So there is one clone of cells which are produced. Suppose we want to produce a monoclonal antibody against a particular antigen or particular target. So what we will do, we will inject that antigen into the mouse. We inject the antigen into the mouse. Then from that mouse, remember that antigen will start producing antibodies. Right? So the B cells are producing the antibodies. So what we will do, we will take the B cells from the spleen of that mouse. The spleen is having the B cells. So those B cells are likely to produce antibodies. So we will isolate the B cells from the spleen of the mouse. Now these B cells, we will fuse them with the myeloma cells. Remember, B cells are mortal. They will divide for 1-2 generations. After that, they will stop. But we want that B cells keep on replicating and they can produce large amount of antibody. Okay. So what we will do, we will fuse them with the myeloma cells. Myeloma cells, they are tumor cells. So basically, they keep on dividing, do not stop. So when we fuse the B cells with myeloma cells, now this cell, this can divide repeatedly. So lot of antibodies can be produced. Okay. So that is the hybridoma technology we use. Then nomenclature. So if we see a normal antibody has a light chain and a heavy chain. So this chain, yellow one is the light chain, whereas the blue one is the heavy chain. This is the heavy chain. Now the heavy chain, it is divided into two parts. It has constant region and the variable region. So if you see, this is the constant region. Till this point, it all is the constant region. So this remain constant in most of the antibodies. But the top portion, this portion is variable. And among this variable portion, the topmost portion, this point is hyper variable region. This is hyper variable region. This is also known as CDR, complementarity determining region. So hyper variable region is there. So this is mainly changing in different antibodies. Okay. So how to keep the nomenclature? The first monoclonal antibody produced was muromonab CD3. So how the name was kept in that was, it was obtained from the mouse. So murine antibody. The name was muromonab. So monab is for monoclonal, AB for antibody. So it is a muro means murine in origin, monoclonal antibody against the CD3 antigen. Okay. So this was used in transplantation. This was the first monoclonal antibody produced. But after that, we decided to keep a particular nomenclature to make the nomenclature easier to understand. So how we kept the nomenclature of monoclonal antibodies is, the name should end with MAB. MAB is monoclonal antibody. And if they are not pure monoclonal antibodies, their name can end with sept. Sept is a fusion protein. Many monoclonal antibodies are not stable. So we need to fuse them with some other compound, only then they become stable. So when they are fusion proteins, their name end with sept, but mostly the name will end with MAB. Okay. Now before the MAB, two letters, they tell the source of monoclonal antibody. That means from where we can obtain these monoclonal antibodies. So we can obtain them either from the animals or we can obtain them from the humans. Or they can be the mixture of the two, some part from the animal, some part from the human. And the mixture can be of two types. One, it can be chimeric. Second, it can be humanized. The first question is, why we want to know from where we have obtained them? The reason is, if an antibody is obtained from animal, foreign component, so there is risk of allergy. More the animal component, more is the risk of allergy. Similarly, if the animal component contains some antigen with that, it can lead to production of antibody against that monoclonal antibody. Yeah? So again, more the animal component, more is the risk of antibody formation. So purely animal monoclonal antibody we do not prefer, because there is high risk of allergy and there is high risk of formation of antibodies against these compounds. So we do not use purely animal. So we use either human or we use mixed. Now mixture can be of two types, chimeric and humanized. Chimeric means 50-50 mixture. 50-50 means the constant region is humans and variable region is animal. So constant region is human, variable region is animal. Whereas humanized means most of it is human. That means constant is human, obviously. And even most of the variable is also human. Only the hyper variable region is animal. So CDR, complementarity determining region, is animal, rest all is human. Now, if it is chimeric, if it is 50-50, then the source code will contain XI. So like we discussed, it is having mab at the end. And then if there is XI here, it is chimeric. Like rituximab. Rituximab. So monoclonal antibody, which is chimeric. Okay. Now, if it is humanized, then humanized, we know humanized, it contains a U. So we have taken U of the humanized. But we saw that U is present in human also. But human did not contain Z. So if we write ZU, it means it is humanized. ZU is humanized. So if we this code, source code contain ZU, it is humanized. Like if we say trastuzumab. Trastuzumab. So it is a monoclonal antibody which is humanized. So ZU means humanized. Then if it is purely human, that it contains U of human. But Z is not there, anything except Z is present, then it will be purely human. Like we have adalimumab. Adalimumab. It's a monoclonal antibody which is purely human. If Z is not there, anything with U, it is purely human. Now, one important thing to remember, nomenclature is not that simple. I have simplified it. There are many complications in that. We have removed that complication just to make you understand. It is not exact. But what we want to know, we want to answer MCQs. So that can be done by the nomenclature I am telling you. Okay. So these are the codes, source codes. But what is important, one question is asked, which monoclonal antibody has high risk of causing allergy, which has lowest risk of causing allergy? So out of the three, chimeric, humanized and human, which has minimum risk of allergy? Yes, it is purely human. So that means umabs have minimum allergy, and which have highest risk of allergy? Yes, it is chimeric, like iximabs. Okay. So this is one thing. Now, before the source, there is presence of a target here. The two letters before the source tell the target. Against which target we have formed the monoclonal antibody. Like if here is TU, it will be for tumors. Okay. So we will discuss the targets in detail in a little while, but we'll discuss some changes. So this nomenclature was made, then the changes were made in that. One change which was required, like adalimumab we discussed. So it is containing the source code as MU. But later on, it was found that the people or the doctors are generalizing that. That means any drug which contain iximab, they will think that it causes very high allergy. But this is not so. It varies from compound to compound also. So the nomenclature in 2009, it removed the source code. So source code is not used after 2009. So like adalimumab was shifted to adalimumab. Source code was removed. Then later on in 2017, what happened? Many companies started making the same monoclonal antibody. Yeah. Like we have generic products, many companies can produce the same compound. But in biologicals, even if same monoclonal antibody is produced by two different companies, it can produce different biological effects. Same antibody, same substance by the two different companies can differ. So how to differentiate monoclonal antibody from one pharmaceutical company from the other? So what they have told that in 2017, that the monoclonal antibody name should contain 4 letters. After the name, there should be 4 letters. There is no meaning of those letters. Just the 4 letters which are specifying that it is of that particular company. So that means the next time the person want to take, the person can take from the same company. So this type of compounds, biological compound which are produced by different companies, which have similar action, they are known as biosimilars. For normal drugs, it is called generic compounds. So in biological compound, it is called biosimilars. Okay. So these are the two changes which are made. Remember, these 4 letters have no meaning at all. Okay. So that is the changes. So now we can move to the targets. Okay. Now the target we discussed here, the mab is present at the end. Then there can be source code or there cannot be the source code. But before this, there will be target. So if the target contains tox in the name, this is for toxin. It is for toxin. So which toxin? We have two drugs, bagilotoxumab. So this is for toxin, and obiltoximab. It is again for toxin. So bagilotoxumab, it is used for pseudomembranous colitis. It is a monoclonal antibody against toxin of Clostridium difficile. Clostridium difficile toxin is neutralized by this monoclonal antibody, and from the name you can remember bagilotoxumab. Low, it is containing low. If I add a little bit C here, it becomes clo. So it is for Clostridium difficile. Similarly, obiltoximab, it is monoclonal antibody against toxin of Bacillus anthracus. Bacillus, that means anthrax. It is against anthrax. So toxin of anthrax bacteria. So you can see it is containing ill. So ill is telling the bacillus illus. So it is monoclonal antibody against bacillus. So no need to remember. If you want to remember, you can remember bagilotoximab, obiltoximab, you can skip. Next is Ne or Nu. Remember, if you write the spelling of neurons, so neurons contain NEU. So if the monoclonal antibody name contains Ne or Nu, it will be working on neurons. Okay. So two important group of diseases which can be targeted by these monoclonal antibodies. One is migraine. The first four drugs, they are monoclonal antibodies used to prevent migraine. So they are against CGRP, calcitonin gene related peptide. They are monoclonal antibody against CGRP. So they are working on trigeminal neuron. So arenumab, eptinezumab, galcanezumab, fremanezumab, they are against CGRP. They are used in migraine prophylaxis. Whereas the last two, which are the new drugs, they are for Alzheimer's disease. They are monoclonal antibody against A beta amyloid, which is depositing in the cholinergic neurons in the brain. So these are monoclonal antibody against Alzheimer's disease. And you can remember aducanumab, the name says for Alzheimer's disease. Monoclonal antibody for Alzheimer's disease. They contain Nu means for neurons, Ne means for neurons. Then these are Nu for neurons, Ne for neurons, Ne for neurons. So Ne or Nu tell about neurons. Okay. Then we have BAC. BAC tells bacteria. So this is a monoclonal antibody for bacteria. So if you see raxibacumab, it is a monoclonal antibody against bacteria. So which bacteria? It is against anthrax. Bacillus anthracus, monoclonal antibody against Bacillus anthracus, or we can say used for anthrax. And again, from the name you can remember, its name start with Rax. So it is used for anthrax. Okay. So this is bacteria. Then next we have VI for virus. If the monoclonal antibody name contain VI, it is for virus. So you can see the first monoclonal antibody, which was only antiviral drug among the monoclonal antibodies since a long time, was palivizumab. So it is a monoclonal antibody. It is humanized and it is used for virus. Which virus? It is indicated in respiratory syncytial virus, indicated in RSV. Then we have found many more drugs, and particularly the most important virus nowadays is, yes, it is coronavirus, COVID-19. So we have monoclonal antibodies against COVID. These are the four drugs: bamlanivimab, imdevimab, sotrovimab, and bebtelovimab. So all of them contains VI means virus. So they are for COVID-19. Then these are the monoclonal antibodies: ansuvimab, atoltivimab, and odacivimab. These are for Ebola virus. Remember, Ebola virus is not common in India. You can safely skip them. And what I advise is no need to remember any of them. Just see if VI is present in the name, and in the exam question is asked where it is used, mark COVID-19. Because COVID is the most important virus nowadays. So any monoclonal antibody they will ask in the exam currently is most likely from the COVID. Okay. So that is about viruses. Then coming to CI, CI means circulation. So if C is present, CI is present, it is in circulation. They will work in circulation. In the blood, it will work somewhere. Circulation. Abciximab, this is the drug you need to remember. Abciximab is monoclonal antibody against glycoprotein 2B oblique 3A. This we discussed in hematology. Yes, it is used as antiplatelet drug. It is used as antiplatelet. The name you can remember, abciximab, CI means circulation, and it is containing A and B, so it is working on B and A, 2B, 3A. Okay. Now next is bevacizumab. In fact, the next three drugs, they are against VEGF, monoclonal antibody against vascular endothelial growth factor, so which is working in circulation obviously. Bevacizumab, CI means circulation. This inhibit angiogenesis. Bevacizumab, it inhibit angiogenesis. And angiogenesis is very important for metastasis. New blood vessels are required. So by inhibiting angiogenesis, it can treat many cancers. So it is used in colorectal carcinoma. It is used in many cancers, renal cell carcinoma, plenty of cancers we can use, non-specific drug. Then ramucirumab, again CI means circulation, CIR you can say circulation. It is against VEGF. So ramucirumab is again anti-cancer, inhibit the metastasis. It is used mainly for gastroesophageal junction cancer and also for non-small cell lung cancer. Okay? So you can remember Ramu was a worker who has habit of drinking and smoking. So because of drinking, he got the problem in the gastroesophageal junction, and because of smoking, he got the lung cancer. So for Ramu, we have gastroesophageal junction cancer and non-small cell lung cancer. Then brolucizumab, it is indicated in age-related macular degeneration. Age-related macular degeneration. So person will not be able to see clearly. Macular degeneration is there. So you can remember, if you say bro, see the Lucy, the person is not able to see the Lucy. So it is brolucizumab. It is for age-related macular degeneration. Then we have caplacizumab. Caplacizumab is a monoclonal antibody targeting the von Willebrand factor. Von Willebrand factor, so it is affecting the platelets. So you can say the name contains PLA for platelets. So it is used in thrombotic thrombocytopenic purpura. Thrombotic thrombocytopenic purpura. Next is emicizumab. Emicizumab is a bispecific monoclonal antibody. It is a bispecific monoclonal antibody. Again, CI means circulation. It bind to factor 9 and factor 10. And after binding, it change the conformation and factor 10 become active. So that can treat hemophilia. So emicizumab is used for hemophilia. Then we have idarucizumab. It is a monoclonal antibody against dabigatran. Dabigatran is an anticoagulant. It is a monoclonal antibody against that. Oral anticoagulant dabigatran. Antidote for dabigatran is idarucizumab. So I will advise you can remember idarucizumab also. Then alirocumab and evolocumab. These are monoclonal antibodies against PCSK9. And they are indicated in homozygous hypercholesterolemia. So this is again important. They have been asked in the MCQs. Alirocumab, evolocumab, you need to remember they are used in hypercholesterolemia. So from the name you can remember they contain OC means over cholesterol, excessive cholesterol is there. Okay, so these are CI or C for circulation. Then we have OS. OS is easy. OS means osteo, means bone. So they will be working on bone. So denosumab is a monoclonal antibody against rank ligand. Remember, this ligand is required on the osteoclast for resorption. Rank ligand must be activated. So we have monoclonal antibody against this, which can prevent osteoporosis. And denosumab you need to remember. This is important drug. Monoclonal antibody against rank ligand, inhibit osteoporosis. Then romosozumab is a new drug. It is monoclonal antibody against a protein called as osteosclerostin. Monoclonal antibody against sclerostin, and this sclerostin, by inhibiting the sclerostin compound, it has dual action, means romosozumab, it stimulate osteoblast also and it inhibit osteoclast also. So by dual action, it is used in osteoporosis. So again, this is also important. Romosozumab has dual action in treatment of osteoporosis. Then third one is Burosumab. Burosumab is monoclonal antibody against fibroblast growth factor 23. It is indicated in X-linked hypophosphatemia. Hypophosphatemia. So basically, what is the function of FGF23 is it help in excretion of phosphate through the kidney. So it causes loss of phosphate. So if there is excessive loss of phosphate, it lead to hypophosphatemia. So we have developed a monoclonal antibody Burosumab against this. So phosphate cannot be lost. So that will treat hypophosphatemia. Okay. So no need to remember this. Just remember Denosumab. And if you can, remember Romosozumab. Okay, next is monoclonal antibodies contain Ki or K. So Ki or K is for kins. Kin means interleukin. See, these are monoclonal antibodies for interleukins. Clear? So all the monoclonal antibody against interleukins, whichever I have mentioned, they are used in psoriasis. Psoriasis. They are used in psoriasis. Ixekizumab is against interleukin 17. Secukinumab is against interleukin 17. Ustekinumab against 2, 12 and 23. Risankizumab against 23. Guselkumab against 23. And Tildrakizumab against 23. So I will not advise to remember any of them. Just see if they contain Ki in the name used in psoriasis. If they are containing Ki or Ku, they are for psoriasis. Okay, no need to remember them. Next is NEB. NEB, these are the drugs which are used for age-related macular degeneration. Nemezumab is against age-related macular degeneration. NEB is for macular degeneration, age-related macular degeneration, neovascularization basically. So ranibizumab is used for ARMD. Again, no need to remember. Next are important. The monoclonal antibodies contain Li or L. So Li means lower immunity. So these are the drug which will lower the immunity. So they will suppress the immunity by, so they are used in either transplantation or autoimmune diseases. So first one, which is most important, is TNF alpha inhibitor. You need to remember that. TNF alpha inhibitors we discussed earlier also. You can remember them as ACE inhibitors को गोली मारो. AC inhibitor को गोली मारो. These are the monoclonal antibody against TNF alpha. A stand for adalimumab. C is sertolizumab. E is etanercept. In is infliximab. And goli is golimumab. So these are monoclonal antibodies against TNF alpha. And these drugs, they are used in rheumatoid arthritis, which is their main use. Apart from that, they are used in Crohn's disease also. And third, they are also approved in psoriasis. And these drugs, they increase the risk of infections. Remember, TNF is protecting from infection. So when we have stopped them, infection chances increases. So they are particularly contraindicated in a patient with hepatitis B or in patient with tuberculosis. Okay. So out of these, all of them are given IV, subcutaneous. All of them are given subcutaneous except infliximab, which is given intravenous. So infliximab name say in means intravenous. Other are subcutaneous. Okay. So that is TNF alpha inhibitors most important. Then we have monoclonal antibody against CD25. The other name of CD25 is interleukin 2 receptor. CD25, it is nothing but interleukin 2 receptor. The drugs are daclizumab and basiliximab. Daclizumab and basiliximab, they are used in transplantation. Monoclonal antibody against interleukin 2 receptor. Interleukin 2 is not able to work, immunity cannot occur, so they are used in transplantation. Okay, if you want, you can remember, otherwise not very important. Then next is monoclonal antibody, which is belimumab. It is against belimumab, again contains Li. So belimumab is against B-lys. B-lys is B lymphocyte stimulator. B lymphocyte stimulator. It is a compound which is overactive in SLE. So belimumab is saying it is BE for B, LIM is lymphocyte. So B lymphocyte stimulator, monoclonal antibody against that, used for SLE. So belly is containing Li, Li means SLE, SLE, Li. So it against SLE. Okay. Again, no need to remember. Next, we have monoclonal antibody against C5. Remember, C5 complement component is overactive in paroxysmal nocturnal hemoglobinuria, PNH. So if we develop a monoclonal antibody against this, we can treat PNH. Okay, and the drug is eculizumab. Eculizumab, you can remember again LI for lower immunity, but C means complement component and E is the fifth letter, A, B, C, D, E. So it is C5. So eculizumab is monoclonal antibody against C5, can be used for PNH, and ravulizumab is the another drug. Again, no need to remember. Next is we have monoclonal antibody against LFA1. LFA1, the drug is ifalizumab. Ifalizumab. Ifalizumab is monoclonal antibody against LFA1. So from the name you can remember LFA, I look like 1. So it is used in psoriasis. LFA is overactive in psoriasis. LFA1 as well as LFA3, they are overactive in psoriasis. So ifalizumab is for psoriasis. Again, no need to remember. Similarly, we have monoclonal antibody against LFA3 and the drug is elifasept. Elifasept. Again, from the name you can remember, it is against LFA. C is the third letter. So it is LFA3. Again, it is used in psoriasis. So again, no need to remember. Next is monoclonal antibody against CCR4. CCR4, the drug is mogamulizumab. Mogamulizumab. It is indicated in mycosis fungoides and Sézary syndrome. Mycosis fungoid and Sézary syndrome, we can use mogamulizumab. Again, no need to remember. Next is monoclonal antibody against alpha-4 integrin and the drug is natalizumab. Natalizumab, it is used in multiple sclerosis. That is important. Multiple sclerosis, that can be asked in the exam. Natalizumab for multiple sclerosis. So what happens in multiple sclerosis? It is a demyelinating disease. Person cannot move the limbs. Person cannot walk properly. So there is demyelinating disease. Neurological problems occur. So if there is a neurological problem, person cannot clap. Obviously, person will not be able to clap. Neurologically, person cannot do these things. So from the name you can remember Natali. Natali. Tali nahi baja payega. Clapping nahi kar payega. Natalizumab. Another indication of this is Crohn's disease. Okay. Next is alpha-4 beta-7 integrin. Monoclonal antibody against alpha-4 beta-7. It is vedolizumab. Vedolizumab. It is used in inflammatory bowel disease like ulcerative colitis and Crohn's disease. Again, no need to remember. Next, we have monoclonal antibodies for asthma. We have 3 monoclonal antibodies which are remembered as OMR. So nowadays it is not used. Earlier OMR sheets were there in which exam was conducted. Example is omalizumab, mepolizumab and reslizumab. Omalizumab, you need to remember, it is a monoclonal antibody against IgE. It is used in asthma. Monoclonal antibody against IgE. Whereas the other two, Mepoli and reslizumab, are monoclonal antibodies against interleukin 5. They are also used in asthma. No need to remember. Just remember omalizumab. Okay. Next is monoclonal antibody against CD20. So this is a new drug, ocrelizumab. It is used in multiple sclerosis. But it's special to remember, it is the only drug which is used in primary progressive multiple sclerosis. Ocrelizumab, you need to remember, it is important for MCQs. Monoclonal antibody against CD20. It is only drug for primary progressive multiple sclerosis. Other drugs are usually used for relapsing remitting multiple sclerosis. Ocrelizumab is for primary progressive multiple sclerosis. Next are monoclonal antibody against interleukin 6 receptors. So first one is sarilumab. Sarilumab, the name tells everything about sarilumab. So sarilumab says it is a monoclonal antibody against interleukin 6. monoclonal antibody against interleukin 6. It is used for rheumatoid arthritis. So everything is written in the name of sarilumab, and this is the drug you need to remember. Then second, also you need to remember tocilizumab. It is also monoclonal antibody against interleukin 6. It is also used in rheumatoid arthritis. Now, second thing to remember, both of these drugs, sarilumab and tocilizumab, they are also approved for cytokine release syndrome. Also approved for cytokine release syndrome. Okay. The next one is crizanlizumab. Crizanlizumab is used for veno-occlusive disease in sickle cell anemia. It is a monoclonal antibody against P-selectin. Okay? So you can see it saying it is used for anemia. A means anemia, C means cell. Sickle cell anemia, crizanlizumab. Okay? No need to remember. Next is monoclonal antibody against CD4. It is ibalizumab. Ibalizumab is monoclonal antibody against CD4. It is a fusion inhibitor. It is used intravenously. IV says IV. It is given IV intravenously. Ibalizumab is for IB. So it is a monoclonal antibody against CD4, and this you need to remember. It is important. Next is interleukin 17 receptor, monoclonal antibody against interleukin 17 receptor A, and it is brodalumab. Again used in psoriasis. Interleukin 17 earlier we discussed is psoriasis, so it is also used in psoriasis. No need to remember. Next is monoclonal antibody against interleukin 4 alpha. 4 receptor alpha. It is dupilumab. Dupilumab is indicated in atopic dermatitis. So you can remember again from the name, D for dermatitis, and D is the fourth letter. So it is against 4 receptor alpha. Interleukin 4 receptor. Again, no need to remember. Next is monoclonal antibody against interleukin gamma. It is empaglumab. You can remember empaglumab is used for primary hemophagocytic lymphohistiocytosis. So it is used for primary lymphohistiocytosis. And Emma look like gamma, so it is against interleukin gamma. Okay, no need to remember. Next is monoclonal antibody against kallikrein. So what is kallikrein? First, in the body there is high molecular weight kininogen which is converted to bradykinin, and the enzyme involved is kallikrein. Kallikrein is the enzyme which convert high molecular weight kininogens to bradykinin, and this bradykinin, if it is excessive, it lead to hereditary angioneurotic edema. So kallikrein inhibitor, the monoclonal antibody against kallikrein is lanadelumab. So lanadelumab is a monoclonal antibody against kallikrein. So it will decrease the formation of bradykinin, so that will treat hereditary angioneurotic edema. Okay, again no need to remember. Next one is immune checkpoint inhibitors. So this topic we have already discussed several places. So quickly revising, this is a T cell. Normally the function of T cell is to kill the tumor cells. So if any tumor cell is there, cancer cell is there, T cell can kill those cells. But problem is T cell can kill a normal cell also. So how to protect a normal cell? So normal cells have been given an ID card. So whenever T cell goes there, try to kill a normal cell, they will show the ID card that I'm normal. So T cell will not kill them. So what is that ID card? So there is an interaction between the T cell and a normal cell, and one of them contains PD1, one of them contains PDL1. So programmed death receptor and programmed death receptor ligand. So basically when this interaction occur, it will inhibit the T cell. Yeah? So this is the immune checkpoint. So normally T cells are inhibited. They do not kill the normals. But cancer cells do not have the ID. So they can be killed by T cells. Now cancer cells came to know that the normal cells have an ID. So they escape. They escape the immunity or escape the degradation by the T cells. So why don't we do that? So cancer cells also start expressing the PD. So when the cancer cells start expressing the PD, so that will lead to again interaction between the cancer cell and T cell. So now T cell will be inhibited and the cancer cells start growing. So that lead to cancers. So now we know that the cancer cells have evaded the immunity by expressing the pseudo ID, false ID. So what we do, we can inhibit this interaction. So if we inhibit this interaction, the T cell cannot be inhibited, so it will kill the cancer cell. Okay, so these are the concept of immune checkpoint inhibitors. Okay, so we are stopping the interaction between the T cell and the cancer cell. Okay, so what are the immune checkpoint inhibitors? You can remember new drugs act at immune checkpoint. These are the new drugs which act at immune checkpoint, and the name of the drugs you can remember: N for nivolumab, nivolumab. D is durvalumab. A is avelumab. AT is atezolizumab. I is ipilimumab. C is semiplimab. P is pembrolizumab. And very recently a new drug has been formed that is called as retifanlimab. Retifanlimab. Okay. So these are immune checkpoint inhibitors. Nivolumab is used for many type of cancers, including the renal cell carcinoma, non-small cell lung cancer. But MCQ which is asked is on Hodgkin's lymphoma. So it is used in Hodgkin's lymphoma. Durvalumab is used for bladder cancer. So you can remember bladder. Dar means bladder. Avelumab is used in Merkel cell carcinoma. Merkel cell carcinoma. So if you remember, there was a South African cricket player called as LB Morkel. LB Morkel. So you can say LB Avelumab look like LV, Morkel look like Merkel. So LB Merkel. So Avelumab for Merkel cell carcinoma. Then atezolizumab is used again for urothelial carcinoma, bladder cancer. We can use atezolizumab. Ipilimumab is used for malignant melanoma. Malignant melanoma, we use ipilimumab. Semiplimab is for squamous cell carcinoma of skin. So you can remember C for cutaneous cancer, skin cancer. Pembrolizumab has very wide spectrum. It is used for almost every cancer: Hodgkin lymphoma, renal cell carcinoma, breast cancer, almost every cancer. But MCQ mostly is asked on EM, means endometrial cancer. Used for endometrial carcinoma. It is also used in Hodgkin's lymphoma. So endometrial carcinoma is the question asked. Retifanlimab is a new drug. It is also approved in Merkel cell carcinoma. So we discussed Merkel cell. Merkel look like Morkel, LB Morkel, and we have lot of fans of LB Morkel. So Fanlimab for Merkel cell carcinoma. Okay, so these are the important drugs. So you can see some of them bind to PD1, some of them bind to PDL1. No need to remember them separately. So nivolumab, pembrolizumab, semiplimab, and retifanlimab, they bind to PD1, whereas atezolizumab, avelumab, durvalumab bind to PDL1, and ipilimumab bind to another antigen called as CTLA4. Okay, no need to remember these details. Okay, now immune checkpoint inhibitors are important. You need to remember. Okay. Now next we have monoclonal antibodies which contain TU or TA in the name. TU or TA is for tumors. They are for tumors. They are for cancers. Okay. So first are growth factor receptors. Monoclonal antibody against epidermal growth factor. Epidermal growth factor. We have drugs like cetuximab. Cetuximab, C for colorectal cancer, C for cervical cancer, that means head and neck cancer. Cetuximab you need to remember. That is important drug. Next is panitumumab. It is against EGFR again, and you can remember start with pani. Pani means water. So when you drink water, it will go to colon. So it will treat colorectal cancer. So panitumumab for colorectal cancer. Then necitumumab is for non-small cell lung cancer and for non-small cell lung cancer. And nimotozumab is a new drug which is used for head and neck cancers and gliomas. So out of these, I will advise you to remember only cetuximab. Cetuximab is important to remember. Okay. Then we have monoclonal antibody against HER2. HER2, we have trastuzumab and pertuzumab. These are important. So these are used in breast cancer. You need to remember them. They are used in breast cancer, trastuzumab and pertuzumab. And if you forget, you can remember trastuzumab instead of T. If we write B, it becomes brastuzumab. Okay, so used for breast cancer, and these drugs can cause cardiotoxicity. These drugs are cardiotoxic. Cardiotoxic. Okay? And you can remember from the name trust. Their name start with the trust. So if you trust someone, heart will be broken. So trastuzumab will break the heart. So trastuzumab is cardiotoxic. Okay. Next is PGFR alpha. So platelet derived growth factor receptor. So the drug against this is olaratumumab. Olaratumumab. So if you remember Brian Lara, which was left-handed, very good batsman. So when he hit the ball softly, it will go very quickly to the boundary. So used for soft means soft tissue sarcoma. So Lara has drived softly, the ball got to boundary, soft tissue sarcoma. No need to remember. Then we have various CD markers and monoclonal antibodies against them. So the latest one you need to remember this, you need to remember CD3. CD3 monoclonal antibodies teplizumab. So you can remember start with the T means 3. Again CD3, it is used for insulin dependent diabetes mellitus. It inhibit the breakdown of beta cells of pancreas. So used for IDDM. Okay. Then we have CD3 and 19. The drug is blinatumomab for Philadelphia ALL. Then CD19 Philadelphia negative ALL. CD19 monoclonal antibodies tafasitamab. It is for diffuse large B cell lymphoma. CD20, this you need to remember, rituximab. Rituximab, any drug containing ritu in the name is against CD20, and that is for B cell cancers like non-Hodgkin lymphoma and chronic lymphoid leukemia. Another CD20 drug is ibritumumab. It is again for B cell lymphoma and chronic lymphoid leukemia. Another drug is ofatumumab for CLL. Tocilizumab for B cell lymphoma, and obinutuzumab for B cell lymphoma and chronic lymphoid leukemia. So these are drugs which are used for B cell cancers. Then another drug is, the latest drug is obinutuzumab. It is for relapsing remitting multiple sclerosis. Relapsing remitting multiple sclerosis. Remember ocrelizumab we discussed earlier, which is for primary progressive. This is for relapsing remitting. Then we have CD22. The drug is inotuzumab. Inotuzumab is for ALL, acute lymphoid leukemia. So what it is saying that I know. So it is saying that say no to I, not me, not I. So what? All is there. ALL look like all. Okay. Then we have CD22. Another drug is moxetumomab for HCL, hairy cell leukemia. No need to remember so many. Just remember teplizumab and rituximab from this table. Next we have monoclonal antibody against CD30, which is brentuximab. It is against Hodgkin's lymphoma and T cell lymphomas. CD33 zemtuzumab is against AML. 38 daratumumab is against multiple myeloma. Isatuximab is against multiple myeloma. 52 lemtuzumab against CLL. 798 79B, it is polatuzumab against diffuse large B cell lymphoma, and SLAM F7 elotuzumab against multiple myeloma. No need to remember any of them. Okay. Then another is TROP2. We have monoclonal antibody against TROP2, which is secukinumab. Secukinumab is used in breast cancer. Secukinumab is used in breast cancer. Again, no need to remember. Then we have monoclonal antibody against IGF-1 receptor. IGF-1 receptor, it is teprotumumab, and this you can remember, teprotumumab, it is used for thyroid eye problem. So it is a monoclonal antibody against thyroid eye disease, teprotumumab. Okay. No need to remember this also. Next, we have Nectin-4, monoclonal antibody against Nectin-4. It is N-fortumab, which is used for urothelial cancer. And again, you can remember N means nectin, N is for nectin, 4 is 4. So it is against Nectin-4, used for urothelial cancer. No need to remember. Next is monoclonal antibody against glycolipid GD2. It is dinutuximab, used for neuroblastoma. So again, you can remember D for D, GD2, and NU for neuroblastoma, used for neuroblastoma. No need to remember. Next, we have monoclonal antibody against B cell maturation antigen. B cell maturation antigen, it is belantamab. It is for multiple myeloma. In Hindi, you can remember, बेलन पड़ेगा तो हड्डी टूट जाएगी. So used for bones, that is multiple myeloma. So these are the various monoclonal antibodies. Okay, so no need to remember all of them, but wherever I say that this you need to remember, try to remember that only. Okay.